Okay, for folks, we're going to talk more about this in the future. But for now, I think it's important that we have some understanding of binding. So there are a couple different theories when it comes to how binding happens. And I think in reality, it's probably a mix of many of these things. The first theory is called the lock and key model. This is where the substrate binds the enzyme and they fit each other perfectly. Think of you putting a key inside of a lock and turning it, right? The key already has a predefined shape that the lock can accept. In contrast, we have something called the induced fit model, and this does involve some shape change that we'll call conformational change. This is when the substrate binds the enzyme in the binding site and some shape change happens. Maybe the binding site gets a little bit tighter around the substrate, or maybe the substrate kind of bends a little bit. The picture on the right here, folks, shows you more of a lock and key model type of binding, where you notice that the substrate is the same shape as the enzyme binding site. You'll notice how this shape here complements the binding site shape here. But it's really more than just shape when we start to talk about binding. So what controls how specific an enzyme is for binding its substrate? Generally, enzymes are fairly specific and will only recognize their own substrate. So yes, shape matters. But as we've learned up until now, folks, hopefully you can appreciate that the types of non-covalent interactions that the amino acids in the enzyme do, they are important. So for sure, we would expect that if the substrate is nonpolar, probably need some nonpolar R groups to interact with that substrate. If the substrate is charged, maybe we want some amino acid R groups that can perform good ionic interactions with that charged substrate. And as a quick note, folks, keep in mind from organic chemistry that molecules can have chirality. So when we're talking about shape, we can kind of just be talking about the overall shape. Is it more circular, more triangular, for example? But we can also be talking about a very specific geometry about any atom in the substrate. And many enzymes are very specific. They will only bind to one of the enantiomers. And so the kind of combination of an enzyme's shape preference, chirality preference, and the types of interactions, non-covalent interactions it can do with substrate, will give us this spectrum of specificities. Some enzymes bind a single substrate, and so they are specific, right? Some are what we call permissive. This is very common for enzymes. Maybe they'll bind 2 or 3 substrates. So for example, on the right here, you see I have pictures of both ethanol and methanol. Well, the enzyme alcohol dehydrogenase inside of our body can bind both ethanol and methanol. And this leads to methanol toxicity because it'll perform a chemical reaction on methanol even though we should not be drinking it. Some enzymes are classified as promiscuous. So this means that the enzyme, it does have a preference for what substrate it binds, but the substrates can be pretty divergent in terms of how they look or what functional groups they have. An example of this is the enzyme chymotrypsin, and you can see its substrates here on the right. So for example, chymotrypsin can perform a hydrolysis reaction on this peptide bond, this bond between a carbon and a nitrogen, but it can also perform a hydrolysis reaction on this ester bond between carbon and oxygen. So even though these are quite different substrates, this enzyme is promiscuous and can bind them both. And so that wraps up our introduction to understanding enzyme binding. Up next, we'll talk a little bit about cofactors to wrap things up.